When we talk about a genome, we often focus on the DNA sequence, the blueprint of life. But understanding what a genome does is a whole different story. This is where we explore things like the transcriptome and proteome, basically, how genes turn into action. Let's start with the transcriptome. This is the collection of all mRNA molecules in a cell at a certain time. These mRNAs tell us which genes are active. We can study the transcriptome by sequencing all the mRNAs, but that's slow and expensive. A smarter shortcut is SAGE, serial analysis of gene expression. It grabs tiny tag sequences from mRNAs, which are just enough to figure out which gene they came from. It's like checking barcodes instead of reading full product labels. Another powerful tool is the microarray or DNA chip. Here's the idea: we turn mRNA into fluorescently labeled cDNA and let it bind to specific spots on a chip that contains DNA from thousands of genes. The brightness of each spot tells us how active that gene is. It's quick and gives us a full snapshot of gene expression in one go. These tools help scientists compare different conditions, like normal versus cancer cells, to see what genes behave differently. In yeast, for example, when sugar runs out and cells switch to a survival mode called sporulation, over 700 mRNAs shoot up and 1,000 go down. It's like a complete software update based on the environment. In humans, these studies help detect cancers. Scientists can even tell the difference between leukemia types just by looking at transcriptome patterns. That's huge for early diagnosis and better treatment. But gene expression is only part of the story. What about the proteins actually doing the work? That's where the proteome comes in, all the proteins a cell is making and using. Studying proteins is tougher than mRNAs. Why? Because protein levels depend on more than gene activity. Some proteins are modified, broken down quickly, or not even active.